Going to be discussing mutations, which is found on chapter 4, section 3 of our books. By the time this section is over, I will be able to identify how mutations can affect an organism, and secondly, explain how cancer is related to mutations in the cell cycle. Your teacher will tell you on what page to write this information in the book, in your journals. And remember, if I get going too fast, you can always pause the video. You can always replay any parts that you didn't get or didn't hear, or if I get going too fast. Let's remind ourselves of the cell. All bodies, plants and animals, are made up of cells. There are, in the human body, 60,000 billion cells, and inside each cell is a nucleus. Inside the nucleus are these X-shaped objects, which we call chromosomes. Chromosomes made up of chromatids. This is a chromatid from mom. This would be from dad. Hundreds of genes on the chromosomes. Here's a blown-up picture of the genes. And of course, genes are made up of DNA. DNA carries the code written with amino acids that make up proteins. So DNA is made up of proteins, and proteins are made up of amino acids. If you think back to the activity that we did with the salamander and the toad, and all those letters that went across the piece of paper, those are the amino acids that we're talking about. In humans, we have 23 pairs for a total of 46 chromosomes in each one of the nuclei of our cells. And that means in this one cell would be over 20,000 plus genes per cell. So number one here, goal, this is what the normal human genome looks like, and it's pretty dang cool. This is what your chromosomes look like. So if you were ever to get this is called a karyotype, which is a picture of your genes. They would go into one of the cells that we just looked at and find the DNA. And as you can see, a lot of them are kind of in the shape of an X. So that's where that comes from. But then they line them up. This is the one from mom, this is the one from dad, or the other way around, and that would be the case all the way through. So here's 22, here's 23, our 23rd pair, which gives us a total of 46. So this is called, the 23rd pair are called the sex chromosomes. These are the ones that decide if you're going to be male or female. This particular one is a male because it has an X and a Y. If you get two X's, then you are a female. And that's how gender is determined in humans. So that's what a normal genome looks like. Chromosomes are made up of genes, as we know. We just read this. There are 20,000 plus genes on all of these 23 pairs. And genes are made up of DNA, of course. So if you do the math, 23 pairs of chromosomes in one cell means that you have 3 billion pairs in all of your cells put together. So lots of chromosomes with lots of traits that we have. So this is still goal number one, and it says mutation. It is in red, and so make sure you get this written down. It's a vocab word. A mutation is defined as any change of the DNA of a gene or a chromosome. Mutations cause the cell to produce incorrect proteins. And this can only be passed on through the sperm and egg. Remember, if you ruin some of the cells that are in your body right now and then have a baby afterwards, you're not going to be passing on whatever change that occurred to your body. So there are three kinds of mutations. The first one is called deletion, so make sure you get that written down. So here's a chromosome, or at least a kind of a picture of one, and what happens is a part of it gets deleted. There's a break in the chromosome, it gets deleted. So you can see this part here, part of the genetic material is absent, and so you end up with a shortened chromosome. So that means everything that codes for these traits here are going to be messed up. They're not going to be normal. The second is called duplication. It's the opposite. So if you have a chromosome here, and these bands, of course, represent genes, if for some reason these two kinds of genes were to get duplicated, you'd have twice the amount of information. Again, your cells wouldn't know what to do with those traits that are involved, and there would be an abnormality that would be produced. The last one is called substitution. And if you think of these letters as the same letters that we had when we were doing the salamander and the toad and the frog activity, remember these stand for proteins here, GAC. And so let's pretend you have GAG and a CTC, and mutation means there's a change. And so instead of having this A here, it would change to a T. Instead of having the T here, that would change to an A. And this actually is an example of a condition called sickle cell anemia, anemia, that we're going to be studying and seeing a movie about. But that's caused by just one amino acid of all of those genes that are made up of DNA, that's made up of protein, and protein's made up of amino acids. One little amino acid mutation, and it causes that very, very awful disease. So get those written down, the 3 different kinds and what they are, what happens. So here is the picture of the sickle cell condition. This would be a normal red blood cell, and as you can see, there's a little cup in here, and that little cup carries a molecule, carries oxygen to all the parts of your blood. And so when sickle cell develops, as you can see, it takes the shape of a sickle. A sickle is kind of a sword knife type item. I think you probably know that. But that oxygen has a very, very hard time lodging into this blood cell and therefore it can't get carried to all the parts of your body. And so there are some symptoms that are associated with that. Of course, the red blood cells are the ones that are affected, and they're affected by having the sickle shape. People are prone to infections, lots of pain, increased risk of stroke, breathing issues. Almost every single part of their body is affected by the lack of oxygen that's being able to be carried to the blood. And this is how they get it. If you have a parent that is heterozygous, and the child was to inherit both little r's, that means that those 2 little r's are going to say that your amino acids, that one amino acid will be mutated, and so therefore it will start producing red blood cells that are in this shape. So there's always a 1 in 4 chance with heterozygous parents. These are considered carriers. They're unaffected. They don't have the characteristics because they have 2 dominant R's. And of course, this guy is not affected whatsoever. So that's how you get sickle cell anemia. This is a disease called Turner syndrome. And as you can see here, with the sex chromosomes, there's only one X. And so since there is no Y, this person is a female. However, and so as you can see, all or you can even have just part of the other X missing. But in this case, it's all missing. The person will probably grow to be pretty short. Body will have lots of swelling throughout their lives. This person will have a very broad chest. The ears will be pretty low set on the head. Person has a webbed neck. Heart issues, increased chance of getting diabetes. Vision is affected. Hearing is affected. Memory is affected. So there's brain issues as well. So that's what happens when there's only one chromosome and the Y is missing, or else the other X is missing. This is one you might be familiar with, Down syndrome. It's pretty common of a lot of syndromes. And what happens is that the 21st pair of chromosomes actually gets duplicated either in part or the entire thing. And so there's 3 21 chromosomes, number 21, instead of 2 from each of those. But with Down syndrome, and it's always the 21st chromosome, growth is impaired. The person has intellectual disabilities and characteristic facial features, puffiness, increased chance of heart issues. And so those are some of the effects of having this duplicated 21st gene as well. So mutations can either be harmful or beneficial. Remember, a mutation is the third way of having issues. For instance, we have an albino alligator here, and so out in the wild, this probably would be pretty harmful, right? He'd stick out. in his environment, and he'd be an easy target as a baby for predators. But put him in a zoo, it's neither harmful nor beneficial, right? Because he's protected there. So here's another example. This is some bacteria. So typically what happens is that we get exposure to bacteria daily. And so you get a disease or, some kind of a bronchitis or something like that. And so you get some bacteria in you. And so this would be the one bacteria, and then the bacteria will multiply. This is typical what happens. So then the infection, we usually get worse when we're sick before we get better. But then what happens is our immune system kicks in, and or some drugs that we might take, antibiotics going to the doctor, is used. And that actually kills the bacteria. So the bacteria die, and then the person becomes healthy again. And so when you're sick, this is what's happening. The bacteria are multiplying until either your immune system and or drugs can get that under control. But there are a number of bacteria out there that have learned to become drug resistant. So again, what happens is the bacteria gets in your body, it multiplies, that's all normal. But then you take a drug, an antibiotic, to kill it, and it doesn't work because the bacteria is resistant to it. So the bacteria will continue to multiply, and when that happens, you're going to have all the symptoms of being sick. So the person remains sick at this point. So this is a mutation that is beneficial definitely for the bacteria, but not for the human. And the reason that these bacteria are able to be resistant to drugs is because when they reproduced, remember the usual scenario with natural selection, a bunch of organisms are born, right? And so we get a whole bunch of bacteria multiplying. Those that might be born with just slightly different characteristics, and those characteristics might contain DNA that contain amino acids that say this code is now rewritten, and so this code says that you're going to like this antibiotic this person's putting in their body, could then lead to that one gets to live long enough to reproduce and pass on those quote-unquote good genes for the bacteria, bad for the human. And the bacteria that weren't born with that slight change in characteristic are the ones that die. But that's okay for the bacteria because there's plenty that are still surviving that were born with that perhaps mutated amino acid that rewrote the code that said you're going to like this drug that the person is taking. So in that case, the mutation is beneficial to the bug, to the bacteria, but harmful to the human. So our second goal was to understand cancer just a little bit. The definition of cancer, it's in red, so please get it written down, is that cells grow and divide uncontrollably and fast. And what happens then is it damages the parts of the body around them. Cancer can be inherited, or the environment can cause cancer, or both. For example, the tar in cigarettes. Tar in cigarettes is an environmental issue. And if you were to have a cancer gene and you smoke, the tar in the cigarettes can augment the situation so that the environment enhances your inheritance of that gene, and then you will start showing the symptoms of cancer, which is for your cells to grow and divide uncontrollably. UV light from the sun or tanning beds can also do the same thing, especially with skin cancer. If you are carrying a skin cancer gene, you may or may not develop skin cancer, but it certainly has increased your risk if you are spending a lot of time in the sun for some people, or spending a lot of time in tanning beds for some people. That UV light can trigger those cells to grow fast and uncontrollably. Now, keep in mind, though, if you may not inherit some of those genes, and that means you can still get these types of cancer from cigarettes. You can create it yourself from the environment or from UV light in the sun, spending too much time there. It's just all depends on who you are and if you're prone to those things or not. So it can be done without inheriting those genes. High-fat diet has also been shown to trigger cells to start growing very fast and dividing uncontrollably. So here's just a picture. This is kind of normal cells and some of the parts, which you're going to actually study in fourth quarter. But over here you can see the cancer cells have a very, very different shape to them. The chromatin inside here is very coarse. There's a lot of cells. They're dividing. This one's in the process of splitting into two pieces right here. The nucleus looks right the way it should. So everything about the cancer cell is not normal to the regular cell. So cancer cells will divide, and so you can see that happening here. They're divide, and then you get this tumor forming, and then, of course, normal cells on either side. And so these cells will then will keep on producing, and then the tumor will get bigger and bigger, and so it will eventually start to affect the cells that are around it, of course, and from functioning properly. So sometimes the cancer will break through this membrane here, and because there's lots of arteries and veins that typically go to tumors, these cancer cells will then be carried through the bloodstream in the veins and the arteries to other parts of the body, in which the whole situation will start all over again, and more tumors can be basically the cells will set up shop in other parts of the body and keep controlling or duplicating uncontrollably. So just a kind of a picture with an electron microscope rather than a clip art type picture. You can see that these little spider fingers that are going to this cancerous cell are all the blood supply, arteries and veins, and so there's lots and lots of ways that these cells in the tumors could break off and be carried away through each one of these arteries to other parts of the body. So cells can be treated by surgery, they can be treated by chemotherapy, or they can be treated by radiation. And so let's go ahead and take a look at a video here, hopefully, see if we can't get that to work, and that kind of describes some of this happening. There we go, it's going to go, I think.
A normal cell to a cancer cell. Take 5 minutes to get the picture. More than 60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes, some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs. These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread, followed by breast cancer, colorectal, stomach, and prostate cancer. The means to combat cancer do, however, exist. These often complementary treatments are used alone or in combination according to the type of cancer and its status. The goal of the treatments is to make it possible to eliminate the tumor and cure the patient in early stage cancer, or like a chronic disease, to control its development. There are mainly 3 kinds of treatment. Surgery consists of removing the tumor in part or in its entirety. Radiotherapy exposes the tumor to radioactive rays, which prevent the diseased cells from multiplying and destroys them. Chemotherapy consists of administering systemic medicinal substances to destroy the cancerous cells or prevent them from spreading. Chemotherapy and radiotherapy act on cancerous cells, but also on healthy cells, which explains their side effects. But today, better knowledge of the characteristics of cancerous cells makes it possible to develop therapies that target the cancer development mechanisms more specifically. Sanofi Aventis R&D is already present in all these areas to fight against cancer on all fronts.
Well, there we go. So make sure you have written in your journals surgery, chemotherapy, and radiation. And if you've forgotten what those are, just back up the video a little bit and write down definitions of those or how those things work. So that's it on our studies of mutations. And other than that,